Hi students, uh, welcome to Biology igcse 610 and we were discussing chapter 5. In the last lecture, we discussed about the basic features of enzyme, and to some extent we also discussed that what is the mechanism of enzyme action. And as I told in the last lecture that next we have to discuss about the factors affecting the rate of enzyme control reaction. That is, enzyme is catalyzing the chemical reactions, but there are certain factors which could affect the rate of enzyme control reaction, either by affecting the structure of enzyme or by affecting the complex formed by enzyme with the substrate, or even they can affect the delivery of product from enzyme substrate after catalysis. So to cut it short, very simple, there are two basic factors which could affect the rate of enzyme control reaction, and one of them is temperature. Let me write it here that one of the factors affecting the rate of enzyme control reaction is temperature. We know that the rate of reaction is directly proportional to kinetic energy. What does it mean? That when substrate and the enzyme, they are colliding more frequently with each other because of their greater motion, greater movements, there is a greater chance that enzyme substrate complex will be formed and the catalysis will occur. So if enzyme and the substrate, they have higher kinetic energy, means they are moving about very quickly, very frequently, then the rate of reaction will be more. And but we also know that the kinetic energy is directly proportional to temperature. That the particles, whether they are enzyme, substrate, atoms, ions, or the molecules, they are moving by getting energy from temperature. That if we will give more temperature, the substrate and enzyme molecule, they will get more kinetic energy. So the sum total of these two relationships is that the rate of reaction is directly proportional to temperature. That if we will increase the temperature, rate of reaction will increase. But the question is, how much will be the increase in rate of reaction for a unit increase in temperature? There is a simple thing, if this is understood to you, that why is rate of reaction increasing by increasing the temperature, then let's discuss about the factor of rise in rate of reaction. There is a law called Q10, or this is also called law of 10. What does it mean? Q10 or law of 10 states that by every 10 degrees Celsius rise in temperature, the rate of reaction controlled by the enzyme is doubled. Means by every 10 degrees Celsius rise in temperature, rate of reaction is doubled. Means the increase in kinetic energy by the 10 degree Celsius rise in temperature is up to such an extent that the rate of reaction is doubled. Now look at this chart. If we are having temperature in degrees Celsius on x-axis with a difference of 10 degrees Celsius, 10, 20, 30, 40, 50, 60 and 70, and we have rate of reaction in arbitrary units, means the number of products formed per unit time or the number of substrate molecules consumed per unit time, that is the rate of reaction. If we are taking rate of reaction on y-axis, and we are here the difference of 2, 2, 4, this is 6, 8, 10, 12, 14, 16 and so on up to 32. I have pointed some points that if at 10 degrees Celsius, if the rate of reaction is 2, then at 20 degrees Celsius, the rate of reaction will become 4. Means 2 to 4, the rate of reaction has doubled. And if we will increase the temperature to 10 degrees Celsius further, means from 20 to 30, the rate of reaction will become 8 from 4. So from 20 to 30, rate of reaction has doubled from 4 to 8. and from 30 to 40 the rate of reaction have become 8 to 16, and from 40 to 50 rate is almost increasing from 16 to 32, but as it is approaching to its denaturation, then it's not increasing up to 32. So if I plot the graph line, it will be like this, it is. Means this is hyperbolic curve, that by every 10-degree rise in temperature, rate of reaction is increasing with the factor of 2. That is, and then after 50 degrees Celsius, the rate of reaction suddenly becomes 0. And we used to say that between 40 and 50 degrees Celsius, it is the optimum temperature. So another word is optimum temperature. I can define optimum temperature in a single word, that this is the best temperature for maximum rate of reaction. The most suitable temperature it is. I'm not saying that at optimum temperature kinetic energy will be in maximum. No, kinetic energy will go on increasing by increasing the temperature even after 50 to 60 to 70. But rate is not further increasing. Maximum rate is at optimum temperature. What happens after optimum temperature? When we increase the temperature beyond optimum, then the 3D shape of enzyme is disturbed. The component of enzyme molecule, they start vibrating with a high frequency, so that the specific 3D shape of enzyme is disturbed. And the 3D shape of the active site of enzyme is disturbed to such an extent that the substrate molecules cannot fit into it. And this stage of the enzyme is called denaturation. So after optimum temperature, there is denaturation of enzyme. That enzymes are denatured after optimum temperature. That's why when temperature is increased beyond 50 degrees Celsius, rate of reaction suddenly becomes 0. Because here there is a very high kinetic energy, but the 3D shape of active site is distorted, so that whether they are colliding with each other very frequently, they are unable to fit into each other. The substrate is unable to fit into the active site. This is called denaturation of enzyme. And this change in the denaturation in the 3D shape of the active site and enzyme is irreversible. That is, once an enzyme is denatured, it cannot be renatured. I can say that this part of the graph is reversible part. I can show you here that I can indicate it with the double arrow, that by increasing the temperature, rate is increasing, and if you will lower the temperature, rate will be lowered. The same Q10 will be applicable, that by every 10 degree rise in temperature, rate will be doubled, and by every 10 degree fall in temperature, rate will become half. But this part of the graph is irreversible. Stop line. Once it is denatured, it cannot be renatured. Means if we have denatured some enzyme at 60 or 70 or even at 80 degrees Celsius, we have distorted its 3D active site shape, and now if we have lowered the temperature of enzyme and we want to let the enzyme work at 30 degrees Celsius, the rate will not be 8, rate will be the same 0. Once it is denatured, it cannot be renatured. This part of the graph is irreversible, while this part of the graph is reversible. Here you have to draw the graph in your ATP papers. You have to draw the graph very carefully, that every point should be clearly marked, every point should be encircled or it is crossed, and then the points should be joined with each other until and unless you are asked to make a freehand line. There should be independent variable on x-axis and the dependent variable on y-axis, because here in this case rate of reaction is depending upon temperature. So temperature is in your control. You are varying the temperature according to your will, and according to the change in temperature rate of reaction is being changed. So rate of reaction is the dependent variable and the temperature is independent variable. Independent variable will always be lying at x-axis and the dependent variable will always be there at y-axis. Hope this is clear to you that by increasing the temperature, rate of reaction is doubled. So one of the factors affecting the rate of enzyme control reaction is temperature. Let's see, there is another factor called pH. There is another factor called pH. What is pH? pH is basically the measure of acidity or alkalinity of a reaction, of a mixture, of a solution, of any medium. It ranges from 1 to 7 to 14. This pH is considered as neutral, this is alkaline, and this is acidic. For every enzyme, there is a specific pH where it is working the best. The most suitable pH for every enzyme is called the optimum pH. And if that optimum pH is changed either towards alkaline or towards acidic, the rate of reaction is equally affected. Here in this case, if we put the pH on x-axis and the rate of reaction as a dependent variable on y-axis, there will be a bell-shaped curve like this. The optimum pH is where the rate of reaction is maximum. This can be any pH for a specific, this can be any pH for some enzyme. For example, for enzyme amylase in our mouth, the optimum pH is about 7. Means at 7 pH, amylase will be working at its maximum, the best it is working. If we will increase the pH towards alkalinity or if we will decrease the pH towards the acidity, the rate of reaction will be equally affected. Basically, the explanation of this effect of pH is that every enzyme, whenever it is synthesized, enzymes are synthesized by the cells, but when enzymes are synthesized, they are made in their specific shape at a specific pH. And if the pH, that specific pH called the optimum pH, is altered, their structure is altered. And when their structure is altered, their efficiency or their rate of catalysis is affected. And the structure is equally affected by increasing the pH or by decreasing the pH. A slight change in the pH will slightly affect the structure of enzyme. And if we will reverse the pH, the same shape of enzyme can be re-achieved. But if the pH is altered up to great extent, then the structure of enzyme will be different, will be altered, it will be changed up to such an extent that it cannot be reversed. So if the enzyme is denatured completely by a drastic change in the pH, it cannot be renatured. It will be destroyed at all. So some part of this graph is reversible. You can say this part of the graph is reversible, while this part of the graph where there are peak changes in the pH is irreversible. And this shape is constant for almost every enzyme, but with a different optimum pH. For example, the optimum pH for Pepsin is about 2. Similarly, the optimum pH for an enzyme in your large intestine is about 8 or 8 to 9, 8.6. But the shape is same, you can see bell shape, bell shape, bell shape. The maximum rate can be different for different enzymes and the optimum pH can be different for different enzymes. But the overall shape is like the bell. There are very few enzymes which are of very strong structure, they are of very strict structure, they are highly resistant to change in pH, so their rate of reaction is not affected by the change in temperature. For them, the graph will be like, you can say the graph will be like this. Just for example, this is, the example of this is papain. Papain is an enzyme which is not affected by any change in pH. So whatever the pH is, their rate of reaction is almost constant. But such examples are very rare. Most of the enzymes are affected by the change in pH and they are equally affected by decrease or by increase in the pH. Hope this is very clear to you that what is the effect of temperature, what is the effect of pH on the rate of enzyme controlled reaction. There are two other factors which could also affect the rate of enzyme controlled reaction. They are the substrate concentration and enzyme concentration. So next we have to discuss that how is the rate of reaction controlled by the enzyme is affected by changing the concentration of substrate and by changing the concentration of enzyme. Then on x-axis, the independent variable will be either substrate concentration or enzyme concentration, but the dependent variable will remain the same, the rate of enzyme control reaction. Hope this topic is very clear to you that what is the effect of temperature and what is the effect of pH on the rate of enzyme control reaction. Thank you.